Hi, I'm Norbert Leitinger.
And I'm Samantha Adamson.
We're from the Department of Pharmacology at the University of Virginia. Recently, my lab became interested in the role extracellular ATP plays in the homeostasis of adipose tissue. However, the mechanisms of how ATP is released from adipocytes are unknown.
Our studies demonstrated that Pannexin 1 channels on adipocytes can release ATP and are also required for full activation of insulin-stimulated glucose uptake in adipocytes. We used cultured 3T3 adipocytes as well as adipocytes isolated from mouse white adipose tissue for our studies. First, we treated adipocytes with known activators of Pannexin 1 channels, and by measuring ATP release, dye uptake, as well as using electrophysiologic recordings, we found that indeed Pannexin 1 channels on adipocytes are functional and can release ATP. Next, we tested insulin-stimulated glucose uptake in adipocytes and found that both pharmacologic and genetic inhibition of Pannexin 1 channels significantly decreased insulin-stimulated glucose uptake. We also found that addition of exogenous ATP to Pannexin 1 knockout adipocytes restored insulin-stimulated glucose uptake. And these findings suggested that insulin itself may be a novel activator of Pannexin 1 channels. So using a heterologous expression system in which we expressed human insulin receptor and Pannexin 1 constructs in HEK cells, we were able to use electrophysiologic recordings to show that indeed insulin is a novel activator of Pannexin 1 channels. To determine whether our findings had any relevance in vivo, we placed wild-type and adipocyte-specific Pannexin 1 knockout mice on a high-fat diet for 12 weeks and measured glucose tolerance. We found that adipocyte-specific Pannexin 1 knockout mice had significantly increased insulin resistance after 12 weeks of feeding compared to wild-type mice. However, the wild-type and knockout mice weighed the same and had the same amount of adiposity, and also we found no difference in the level of adipose tissue inflammation. To determine whether our findings extended to humans, we measured Pannexin 1 mRNA levels in human visceral adipose tissue and surprisingly found that Pannexin 1 expression levels were positively correlated with the degree of insulin resistance, suggesting that Pannexin 1 may also play a role in human insulin resistance.